So, hello everybody, good morning, good afternoon, good evening from wherever you are sitting. I'm Martin Friede at the World Health Organization, where I lead the Vaccine Research Unit, and I'm a member of the ADVAC Scientific Committee. So today we are going to be having a seminar on mRNA, and I think this has attracted a huge amount of interest. We've got over 350 registered participants. For the moment, there are 109 that have already joined us. Hopefully some more will be joining as we go through this. So just a bit of housekeeping. We're going to have two presentations first, and we're then going to open up these to questions and answers. So you can all see your chat box. Please put your questions into the chat box. You can put the questions in during the talks, put the questions in after the talks, and I will then, when we finish these talks, I will call on you to open your camera so we can see you while I read out your question. The this meeting is being recorded. The recording will be available online after this call. So today we're going to be having two presentations. The first one is from Barney Graham. I'll introduce him in a moment, and then from Krause, whom I will also introduce. These are two really eminent speakers. We're delighted we've been able to get them to join us today. But I'm first going to just say a few words about why did we choose mRNA as the topic for today. And from the 350 participants registered, clearly this topic got a lot of interest. So therefore, I don't have to remind you of the huge impact that mRNA has had on COVID response. Prior to COVID, mRNA vaccines were a rather obtuse topic that were being discussed in a few vaccine conferences, and a lot of people were interested but mildly skeptical. So this huge benefit that mRNA showed for COVID in terms of the speed at which we were able to develop the vaccines, the adaptability, and then looking at this and saying, well, this could really change the way that we make vaccines. This has really created a huge demand from many countries who said, we want mRNA technology too. Now, in response to this call from our member states, from countries to say, we want the mRNA technology, which is currently in the hands of primarily BioNTech and Moderna, but many biotech companies are also developing mRNA technologies. WHO has established an mRNA technology transfer hub. This is, again, if you look on the web, you'll find a lot of information about this. This has been established in South Africa. So in South Africa, the technology is being established so that it can be shared afterwards with other manufacturers in other low and middle income countries to enable these countries to establish mRNA vaccine technologies themselves. And you will also have seen reports from BioNTech and from Moderna about their intentions to also be building factories in low and middle income countries. But one of the big challenges that we have when looking at these plans to do this is, well, mRNA, it's been great for COVID, but if we build these factories, what are we going to do with these factories after COVID? And so hopefully Barney will be able to give us some insight into what potentially we can do with mRNA other than COVID vaccines. And then the other big questions that we're getting from groups that are looking at setting up mRNA is saying, well, how are these vaccines going to be approved? And how are we going to even approve COVID vaccines as we go further into the COVID outbreak? And everybody will either have been infected or will have been vaccinated. And how are we going to approve the variants as we make vaccines against the variants coming along? So hopefully Krause, who's this is really his life, will be able to give us some guidance on how manufacturers and countries will need to be thinking about this going forwards. So that's why I'm going to stop my introduction, and so it gives me a really huge pleasure to now introduce to you Barney Graham. So Barney, for all of you that have been in ADVAC, I'm sure you will know Barney. Barney's very well known to those of us in the vaccine research and development field. Barney is the former deputy director. When we say former, I think he left this role only a few weeks ago. He was the former deputy director of the NIAID Vaccine ResearchCenter and has developed novel vaccines and monoclonal antibodies for coronavirus.
Okay, it seems this is Philippe Duclos. Apologies, it seems that we lost temporarily our chair, Martin. Hopefully he'll join back very briefly.
You'd like me to start, Philip?
Let's give him 1 minute chance to come back and finish his introduction of you, and then you can get started if he doesn't connect again. Okay, it seems that he's having challenges to reconnect. I'm going to call him and see what's up. And perhaps I would suggest then that Barney, you get started. I think he said the most important about you, and for most of the ADVACers, you need actually no introduction because you lecture year after year, and they all know you pretty well. And your reputation and expertise in mRNA is basically well established. So please get started. I'll try to reach out in parallel to Martin to see what's up.
Okay, well, good morning and good afternoon and good evening to everyone. I am Barney Graham, and I'm former Deputy Director at the Vaccine Research Center. I retired from federal service at the end of August, but hope to remain active, and I'm very glad to be giving you this, my thoughts about mRNA today. I have these disclosures. I'm an inventor on vaccine and monoclonal antibody patents for a number of viral diseases, but nothing specific to mRNA. And the history of mRNA goes back to the 1960s. And since then, a number of groups, a number of organizations have tried to apply mRNA technology to therapeutics, either for vaccines or for treatment of various disorders. And the history also includes a number of findings of how mRNA itself is especially viral RNA, is an activator or an inducer of inflammation and immune responses. So the mRNA I'll be talking about today, these are usually delivered, encapsulated in a lipid nanoparticle. I'll say more about that, but when mRNA goes into the cell, especially viral mRNA, it can trigger TLR7 that sees an uncapped single-stranded RNA, or it can trigger TLR3 as double-stranded RNA, or it can trigger intracytoplasmic receptors like RIG-I and MDA5 in the cytoplasm as double-stranded RNA. And these all lead to the same thing, which is induction of type 1 interferons and pro-inflammatory cytokines. So part of the mRNA biology is induction of inflammation, and the level of induction of inflammation can affect how the mRNA works. And so the types of RNA vaccines and mRNA vaccines include what would be called conventional mRNA, which is simply adding a 5' cap to avoid some of the TLR7 signaling, expression of the antigen of interest, and then the poly-A tail, as opposed to self-amplifying mRNA or SAM, which uses the amplicon from alpha viruses. And so it has a cap, but it also has the some of the internal proteins of the alpha virus amplicon to express multiple transcripts from each RNA cassette. There's also a lot of choices you have to make about how this RNA is constructed. So the 5' cap is essential, as I mentioned, to reduce TLR7 signaling. And the open reading frame is something we'll talk about in a minute, but there's a number of things you have to decide in the untranslated regions of the RNA that we've learned a lot about from flavivirus biology. So 5' untranslated or 3' untranslated regions can affect secondary structure of RNA and expression of RNA, and therefore expression of the protein. You also have to think about whether you want modified nucleotides or unmodified nucleotides. Mostly in this case, it's meant pseudouridines or 1-methyl or 5-methyluridines can be added into the sequence, and that reduces some of the induction of these type 1 interferon signaling responses. Codon modification is critical, as it is in DNA manufacturing, and I think we need a lot more information about that. Leader sequences can affect the overall translation efficiency from the transcripts, and secondary RNA structure can affect translation. And then there's formulation questions that can affect some of the initial, not only preserving the integrity of the RNA, but some of the initial innate immune stimuli. And all of the RNAs are characterized by packing into lipid nanoparticles. And these contain 4 different types of lipid: an ionizable cationic lipid, cholesterol, a neutral lipid, and pegylated lipids in various combinations, and slightly different versions of each lipid are present in each different manufacturer. For instance, in Pfizer-BioNTech and Moderna lipid nanoparticles, these vaccines for COVID are almost identical. They both use 1-methyluridine, pseudouridines in the manufacturing. They both have these 4 types of lipids, but 2 of those lipids are different. The pegylated lipid is a bit different, and the cationic lipid is a bit different. So these can affect some of the early innate signaling processes that can affect them, either the immunogenicity or the reactogenicity of the product. So I've made these charts. We're in a unique year. This last 2 years in vaccinology, we have had the opportunity to test vectors, nucleic acid delivery, protein-based approaches, and whole inactivated virus, and take things all the way through to efficacy testing. And testing multiple modalities against usually a very similar antigen, and looking at that kind of data for both side effect profiles and efficacy is really unique in the history of vaccinology. And it has told us some things that I think we can use going into the future. And so what I've done is list the different types of modalities of vaccines across the top, and some of the things that I think would be pros for the modality in green, and negatives for the, or cons for the different modalities. And I think what you can see right off is that RNA is pretty positive in the pros and pretty negative in the cons. And so it fulfills a lot of these benefits, and that was before. And now after this last 2 years, we also have a licensed product in mRNA. And we've learned that the stability, it can be managed and it can also be improved. And so virtually every positive box is checked and the negative boxes are relatively unchecked. And so the great advantage of RNA is rapid manufacturing, but it can also induce antibody. It can induce CD8 T cells, which is they are not induced by proteins and whole viruses. It can induce CD4 Th1 type T cells, which are also induced by DNA or vector-based approaches, and maybe less so by whole inactivated or protein-based approaches. And somewhat uniquely, it induces a potent T follicular helper cell response that we think may be important, as demonstrated by Drew Weissman's group. Some of the disadvantages of some of these modalities, like when the nucleic acid of the vaccine has to go into the nucleus to be active, as it does in many vectors and DNA, that's not the case in mRNA. So I think that diminishes some of the theoretical concerns. Rare adverse events, as these vaccines have rolled out, we've seen happen in some of the vectors. There's a few things that happen in the nucleic acid vaccines, and proteins, I think, are relatively safe compared to these whole inactivated products. Unlike the vectors, RNA doesn't have anti-vector immunity, which allows it to be used repeatedly. And it also avoids these Th2 type responses that we'll mention later that could potentially be a problem for respiratory virus immunity. And importantly, and we'll get to this at the end, it does not require mammalian cell culture or bioreactors for its manufacturing process. And being a synthetic chemistry manufacturing approach, I think it could be a game-changing fact. So this is a slide adapted from Miles Davenport from the University of New South Wales. And what he's done is look at the serology from all these different efficacy trials that have been done with either mRNA proteins, advectors, or whole inactivated vaccines, and ask where do those vaccines fall on the scale of relative immunogenicity for neutralizing activity compared to convalescent sera. So convalescent sera is at the bottom, and he provides this relative efficacy grade, which at the 50% level is this whited out space. And so 50% protection is occurring at this level of neutralizing activity. And so he can create a hierarchy of efficacy looking between platforms. And you see that mRNA or protein or even the advectors can create a large efficacy, especially 2 doses of the Ad26. And I've starred the products here that have stabilized spike proteins, which preserves some of the neutralization sensitive epitopes, and they also improve protein expression. But you see that if you have mRNA or protein in stabilized form, you're getting a very high level of efficacy induced. And that is why everyone's excited and interested in RNA. And so the reasons we chose RNA as part of our pandemic preparedness program are because those immunological properties, but in part because we wanted to avoid any prospect of vaccine-enhanced illness that we have studied for a long time in respiratory syncytial virus. And And the properties of the vaccine-associated enhanced respiratory disease are that you have a poorly functional antibody, which in this case is accommodated mostly by the antigen design and making sure the quality of the antibody response is high, but also to avoid this Th2 biased response. And when you have a Th2 response and increased IL-4 or 13, for instance, increases the inflammation and impairs cytolytic function of CD8 T cells, it can generate this neutrophilic alveolitis, which is characteristic of the vaccine-enhanced illness. And it can create mucus production, especially from the IL-4, and airway constriction from the IL-13 in particular. And so avoiding these kind of things that narrow airways or creates inappropriate or ineffective inflammation or narrows airways from constriction is something you want to avoid. And so using a nucleic acid vaccine that induces CD8s and Th1 bias is one of the main reasons we chose RNA, beyond the fact that it's very rapid manufacturing. So going into the future, now that it's a proven technology, the question is, how is it going to be used? And I've listed some of the ways here that it will be used. You can express individual proteins, as we did with the coronavirus spike, or that is being done with a respiratory syncytial virus vaccine that's being developed by NIPA. It can also be used as a multi-component vaccine because it can express multiple RNAs at the same time. For instance, the CMV vaccine that Moderna is developing has all 5 components of the pentameric piece and the GB molecule all expressed in the same set of nanoparticles, and a similar program is going on for EBV. You can make these vaccines and antigens, especially the class 1 fusion protein antigens, either membrane anchored or secreted. The spike protein and the RSV F protein vaccines are membrane anchored, but the NIPA vaccine, it's not drawn right here, but it is designed to be secreted so that it can express both the F and G components, or antigenic components. We also know that it can be a vehicle for launching particle-based or self-assembling particles like the chikungunya alpha virus particle or the Zika VLP. It's being used for cancer vaccines, even personalized vaccines, because of rapid manufacturing. It can be used to deliver monoclonal antibodies, and it can be used to deliver combination vaccines, as some groups are doing for a multi-component or multivalent respiratory vaccine that might include influenza and coronavirus, RSV, and HMPV, for instance. So there's going to be a lot of uses for RNA going into the future. And for pandemic preparedness, the way we've organized it in our own minds is for these 26 viral families that can infect humans, you can divide them into different mechanisms of entry. So class 1 fusion proteins like RSV or the coronavirus spike, we now have evidence that mRNA can effectively deliver these kinds of vaccine antigens. We have evidence that it can deliver flavivirus antigens in the class 2 viruses and herpes virus antigens in the class 3. And now with EVD68 and some of the others, there's evidence that it can also induce or deliver these particle-based vaccines. So mRNA has a lot of potential for delivering a lot of different types of vaccine antigens. Now, I just want to make a point now that the promise I see in RNA is a way of advancing manufacturing into low and middle income countries. And I'm going to make an analogy to cellular technology, not biology, but cell phone technology in Africa, and how just 20 years ago, there was a large disparity in access to cell phones and communication equipment between Western and African countries. But with the introduction of cell phones, that gap has narrowed greatly. And it's because Africa didn't just invest in communication technology, it basically jumped over a couple generations of cable to make a cell phone technology that allows all these different things to happen. It's activated the economy, it makes money transfers possible, and all sorts of information that really has improved life overall. And in the same way that moving from cable, directly using new technologies to satellite and wireless, you can envision that going from bioreactors on large campuses to make vaccine products to something that has a small footprint, small batch capacity for making RNA in a chemical synthesis in modular facilities that could be used for clean rooms and GMP, then this technology really has the capacity to move into low and middle income countries and change the whole paradigm for how we manufacture. So mRNA rapid manufacturing by chemical synthesis is a platform technology that allows the process to be perfected in certain ways and to be done in a similar way each time that hopefully will streamline the regulatory process that you'll hear about from Dr. Krause. It can potently induce CD8 and antibody and promotes Th1 and T follicular helper cell responses, which I think are the type of response I would prefer for a vaccine, a respiratory virus in particular. The data now we have in hundreds of millions of people suggests that mRNA can be delivered safely and it's efficacious. If it doesn't have anti-vector immunity, the stability of the product and the supply chain has greatly improved over this last couple of years. The fact that it's a small footprint, small batch manufacturing approach is well suited for low and middle income countries and regional problems. And there are a lot of things that need to be done. I think that there's room for improvement in how we select codon usage, the secondary RNA structure, and its impact on expression and translation efficiency, I think, needs a lot of work. The downstream processing, especially to removal of double-stranded RNA, which can still activate some of these inflammatory pathways, is important. The lipid composition and formulation can be improved, and delivery, especially to mucosal surfaces, I think could make another big advance for mRNA. And I just want to end by saying that mRNA is not magic. People have gotten very excited about this, and I am also hopeful about its potential, but it won't work if you don't deliver the right antigen. And I just want to emphasize how critical antigen design is, both from a structural basis at the atomic level detail to get antibody responses right and high quality antibody, but also at the particle level, at the quaternary structure level, that I think we will appreciate the importance of antigen design as time goes forward, because RNA is not going to work for everything if you don't deliver the right antigen. So I will stop there and take any questions if you have. Or we can all go to Philip's talk and do the questions later.
Thank you very much, Barney. That was really excellent, and it gives us a lot of hope going forwards. I'm going to suggest that we keep the questions until after Phil Krause's presentation. And so to all the participants, please, if you've got questions, put your questions into the chat. Okay, but we will open for questions after the second presentation. So thank you, Barney. So it's now really my pleasure to introduce Phil Krause to you. So Phil Krause has been the Deputy Director of the Office of Vaccines Research and Review at FDA for the last 10 years. Before that, he served in various other capacities at the FDA. Now, for those of you that have been working even in conjunction or related to WHO's many activities regarding the Ebola outbreak, the R&D blueprint, and more recently, of course, the COVID response, you will have bumped into Phil giving advice to WHO and to the community at all of these meetings. He's been a real pillar providing support to the community on how to think about the regulatory approval process for all of these products, not only vaccines, but also monoclonal antibodies for application in these infectious diseases. So Phil, please, Philip, over to you, please.
Thanks. Are you able to see this slide? It's the ADVAC slide, and hear me?
I can hear you.
Good. Okay. So I'm repurposing the main theme slide here as my title slide as well. And I've focused in this talk fairly narrowly on this question of whether the success of mRNA vaccine development platforms for COVID will lead to new platforms for old vaccines, which I take to mean that whether mRNA will be a useful modality to replace some of the old vaccines. But I think that there are very interesting questions about newer vaccines and where to go with COVID with mRNA vectors, which hopefully we can get to in the discussion. So let me see why I'm not able to move forward. Okay, I'll start with a disclaimer, which is that I have no conflicts to report, but that this talk represents my own personal opinions, which aren't necessarily those of any organization or any disorganization. Oops. So when I talk about regulations, I often start with this slide, which sort of describes how I like to think about regulations, which may be different from how many of you do. Regulations do have the force of law, but we want regulatory decisions to be based on good science. And so for vaccine development, this implies that study designs will provide scientifically valid results. That is, the studies are adequate and well controlled. They'll conform to ethical and human subjects protection standards, and likely will yield outcomes that could ultimately support broader availability of vaccine, ideally via licensure. And this, of course, implies the importance of early and frequent conversations with regulators to help assure that the study designs and objectives will meet the goals. But the other thing this means is I don't like to talk about this concept of a regulatory challenge, because I don't think there's a such thing as a regulatory challenge. I see everything that people would otherwise consider a regulatory challenge to be a scientific challenge. And to the extent that a regulator disagrees with a developer, it means that there's a disagreement over the science, because I think the regulations merely put into law the thing that everybody's trying to do, which is get products out there which work and are safe. And so to me, if a developer is doing what needs to be done in order to justify the claims they would like to make for the product, they shouldn't have many problems with regulators. But we'll talk a little bit more about some of the details of the regulatory pathways in a moment. So Barney has gone over mRNA vaccines very, very well. And I wanted to start, though, with a few potential advantages of them. Obviously, they're relatively easy to make and potentially even adjust, as we saw when the manufacturers were able to, for instance, fairly quickly make and start testing in humans beta variant vaccines. Previous experience with an mRNA vaccine surely could streamline movement into phase 1. Indeed, for COVID, previous experience with other mRNA vaccines that had been in the clinic, although obviously not licensed, was used to justify moving very quickly to phase 1. So there is some advantage in terms of moving quickly into clinical trials. And of course, nowadays, we think of phase 1, but very often these first trials are phase 1/2 trials and provide substantial immunogenicity and dose finding information, such that I think with mRNA vaccines, there is an advantage that in many cases it will be possible to move very quickly to getting data which will help define what the final product should look like. They certainly give rise to high antibody titers. I think that these high antibody titers, along with what, at least for COVID, appears to be a high potential likelihood of efficacy, might support more at-risk development decisions by manufacturers, where they might be willing to go forward based on less information. Because, of course, a lot of the information that is collected in early stages of development isn't just to satisfy regulators, it's to satisfy the developers that it's worth investing the amount of money that it would cost to do phase 3 studies. And so it may be there's an advantage there. And there are also some cell-mediated responses, although I'll note is that these might not always be to the most important antigens for cell-mediated immunity. At least for many viruses, it's not the surface proteins or glycoproteins which induce the greatest cell-mediated responses in humans. And so if one is counting on an mRNA vaccine to induce a cell-mediated response, then one ought to think again about what are the right antigens for doing that. What are potential disadvantages? Well, there are some questions about durability of protection as manifested in the recent booster debate in the US, where it appears as though there may be some waning of protection, although in my opinion, not a lot, and that protection against severe disease with COVID has actually been very well retained, which doesn't, of course, mean that one wouldn't give boosters to people who are at risk for very serious disease, even with 2 doses, but nonetheless, some of these concerns might be overblown. And yet it does seem as though, well, there are some questions about durability of protection, and it's unclear what the regimen needs to be in order to get the best protection. And for instance, even now for COVID, we've used 2-dose regimens. Would we contemplate a 2-dose mRNA vaccine regimen for influenza? Or if we ultimately decided we needed 3-dose regimens, would that be the right way to go? And of course, we also don't know what the right pediatric regimens of these vaccines are. It's still very early in their development, and finding something that will work for COVID doesn't necessarily mean that we figured out the right dosing and regimen for other diseases. I think the role of original antigenic sin isn't yet that well defined. And for this, I wonder for viruses that change over time, whether the degree to which an early mRNA vaccine response then can be overcome by subsequent vaccination with different antigens, and whether that ends up being an issue. And as Barney pointed out, we really have to be sure that we've got the right antigen in order to get both protection and durability of protection. Safety, of course, there are some potential issues, possible autoimmune risks, especially if we're contemplating giving people many doses of mRNA vaccines are going to be used for many different diseases. Myocarditis was more common after the 2nd dose, although appeared to be fairly self-limited in most people. And so it's unclear whether that's really an autoimmune phenomenon or not. But that's something to keep an eye on. And there may be other potential risks associated with frequent re-exposure to the same modality. The lipid nanoparticles, for example, are not things that people are that frequently exposed to parenterally. And so there may be some implications of many vaccinations there. And there is, of course, always heightened safety concern for pediatric vaccines. I'll add another consideration, a couple other considerations just for research, and that is pointing out that lipid nanoparticles likely do play a critical role in the immune response. The methylpseudouridine manages to allow the mRNA, if it gets into cells, to avoid the cellular defenses, pattern recognition receptors, CGAS. Well, for DNA, it would be CGAS and STING. For RNA, probably PKR, and so forth. But in doing that, it's not clear that without the lipid, which is also given along with this RNA, we would get such a strong immune response. Because the lipid itself does play a role in recruiting inflammatory cells, And it's at least my belief that you need to have both the RNA and the right lipid nanoparticle. And so figuring out what are the right and best lipid nanoparticles may be an area for additional research, which may change and give us a mechanism for further improving these vaccines. And then one could ask, if one is asking a question about the lipid nanoparticles, one could also ask whether there are other adjuvants that could further increase the immune responses or duration of protection and make mRNA vaccines even better and more suitable for certain diseases. So since the question we're asking and answering here has to do with old vaccines, I guess the first question then, if one is going to make an mRNA vaccine for that, is there a market for a replacement in the first place? Is an mRNA vaccine likely to be cheaper and somehow better? And second then, is an mRNA vaccine likely to work against the pathogen targeted by the old vaccine? So what I did was I went through the list on the FDA website of all of the US licensed vaccines and transcribed them by hand. So hopefully I didn't miss any, but I think this is a fairly complete list to think about what are all of the current old vaccine targets and what are the antigens that are currently used in order to vaccinate against them if we're thinking about replacing these old vaccines. And for bacterial vaccines, what you can see is that these absorbed vaccines all involve killed pathogens. There are a few live pathogens. And of course, there are a lot of pathogens where the vaccines are actually polysaccharides, in some cases conjugated. And so one wouldn't be able to manufacture a polysaccharide with an mRNA vaccine. So for the bacterial vaccines, the only currently licensed vaccine that is recombinant and thus might be a simple target for an mRNA vaccine where one could take, in this case, the several meninge group B antigens that are recombinantly provided and encode those in mRNAs. But all of these other vaccines include substantial numbers of additional antigens which one would not easily be able to encode with mRNA. So meninge group B, at least on the basis of it being a recombinant vaccine, might be an old vaccine that one could consider a target for replacement with mRNA vaccines. And here's my list of the viral vaccines. And here, of course, it's a little bit more promising, but we do have a lot of live vaccines, of course, in the viral space. And it seems unlikely that live vaccines, or in the case, for instance, of rotavirus, where the vaccine probably also causes some mucosal immunity, and is also a live vaccine, that one would be able to get the full range of antigen presentation with an mRNA vaccine. Now, there is one exception to that, and that is now I don't see it on my list here. So, but is herpes zoster, which is of course varicella. The live attenuated varicella vaccine became a zoster vaccine, and that zoster vaccine was a live vaccine. But the Shingrix, the GSK vaccine, of course, presents only glycoprotein E, which is the predominant glycoprotein on the surface of VZV. But nonetheless, that's a situation where we do have a live vaccine that was replaced with, in essence, a recombinantly produced vaccine. And so that doesn't mean that it's impossible, but it does mean that perhaps it'd be more difficult and require some more thinking about what exactly the vaccine is, mechanism of action is, and what antigens it's really using in order to lead to protection. So if you look then at the vaccines on this list that are recombinant, however, we have hepatitis B, which surely seems like that could easily be encoded by an mRNA, although it may be that there's not great demand for an mRNA hepatitis B vaccine. There are some improved hepatitis B vaccines out there with different adjuvants to try to target people who are not responding well to the original one. And yet that's a fairly small market, I would think, for an mRNA vaccine plant. Human papillomavirus, of course, is recombinant. And there one could imagine, although if one wanted to protect against a lot of HPV strains, making sure that one is getting the ratios of the mRNAs right where one would presumably be having all of these antigens produced in the same cells might be complicated, but that might then be a target. And then the other recombinant vaccine on this list is influenza, where of course there are inactivated egg-produced vaccines, there's live cold-adapted vaccine, and there's also recombinant vaccine that's made in baculovirus. And so, and of course, the inactivated and live vaccines are presenting many antigens, but here also the recombinant vaccine is hemagglutinin only. And so it does seem like influenza might be a reasonable target for an mRNA vaccine. I'll add that there, if I were going to pick additional targets from this list, I would think about some of these flaviviruses, JEV or maybe TBE, where it seems like very small quantities of antibodies are useful for protecting against them. We have a reasonable sense that neutralizing antibodies are a correlative protection, and it's not too difficult to induce them. And so those might be reasonable targets as well for mRNA vaccines. But this is heavily opinion-based. So let's say we've picked our target and we want to license the vaccine. Well, how do we get there? Well, first off, we need safety data, and that's what's in red on the slide. So there's no difference among these pathways based on whether that differs in the amount of safety data that's needed. It considers the nature of the product, its intended use, and the severity of disease to be prevented. But there are differences in these pathways depending on the circumstances and specific eligibility criteria, especially for accelerated approval in the animal rule. Whoops, I'm going the wrong way here. So traditional approval normally is based on pre-licensure clinical studies that can provide evidence of effectiveness based either on protection against clinical disease, but in some cases where there's a scientifically well-established immunologic marker to predict protection that can be reliably measured in a validated assay, an immunologic response can allow you to go directly to a traditional approval. If a traditional approval, so normally accelerated approval is done with the idea of getting a vaccine out earlier based on a surrogate endpoint that's reasonably likely to predict clinical benefit, but with a requirement to later verify clinical benefit through required post-marketing studies, which are usually underway at the time of approval, must be adequate and well-controlled, and must be conducted with due diligence. But accelerated approval does provide a way to get a product on the market, at least initially, based on immune responses. And if one is thinking about old vaccines and being able to compare the immune response from an mRNA vaccine with an old vaccine, if it seems like one might be able to use those kinds of comparisons to come up with results that are reasonably likely to predict clinical benefit. But of course, there still is a lot of discussion about exactly how this verification can go. When accelerated approvals first started being done, there was a sense that this verification ought to be done with randomized trials, which were already underway. But increasingly, it's becoming appreciated that there may be some vaccines for which there wouldn't be any licensure pathway besides accelerated approval, if such a study can't be done, a clinical study can't be done, with then confirmation using some kind of post-marketing observational studies. And so that's a topic of intense interest and discussion, which I won't say any more about, but may provide a mechanism also for moving some of these mRNA vaccines forward. And then, of course, is the animal rule, which is products that have been studied for safety. There's my animal speaking on cue. Studied for safety and efficacy in ameliorating and preventing serious or life-threatening conditions, where there has to be an impossibility of approving the product based on one of these other efficacy standards, with a requirement for follow-up studies when feasible to verify benefit. So the number of cases where we have old vaccines where an animal rule would apply, I think, would be practically zero. Now, there is a very interesting influenza vaccines guidance that was put out about 15 years ago, describing clinical data needed to support licensure of seasonal inactivated influenza vaccines. And it discussed approval via accelerated approval of flu vaccines using placebo-controlled immunogenicity studies and set out specific criteria that needed to be met. And this included supply considerations. And here, again, it was stated that the accelerated approval could be based on non-inferiority versus a licensed seasonal influenza vaccine. And then also set out specific seroconversion and GMT seroresponse criteria, which could also permit an accelerated approval. So this is a table on the next 3 slides, and I'm not going to go over it in great detail, but I wanted to copy it into the presentation because I imagine this will be shared with all of you, which describes different surrogate endpoints which have been used for various regulatory decisions with vaccines. And these are also available on the FDA website. But you can see there are a lot of vaccines for which antibody responses have been used and have been thought of as surrogate endpoints for one or another purpose, not necessarily the original licensure, sometimes for bridging to additional groups and so forth. But this sort of creates a starting point for looking at the ability to use accelerated approval or to think about accelerated approvals for some of these vaccines. And so here we have, of course, Hep A and Hep B, and flu vaccines. I described the HAI antibody, but even and JEV I mentioned a neutralizing antibody. Meninge of all types has been approved based on serum bactericidal antibodies, and then there are other vaccines where neutralizing antibody responses have also been used. And here is the website where you can download this table together with also a lot of other drugs for which surrogate markers are also listed. So to conclude, I think for the old vaccines, I think the diseases that can be prevented by recombinant vaccines may be most amenable as mRNA vaccine targets. And I would add to that some of these flaviviruses with some more skepticism about dengue though. And then for many pathogens, mRNA vaccines could potentially be evaluated by a non-inferior immune response to existing vaccines, where the existence of a previously licensed vaccine might provide some additional simplicity for getting you to an accelerated approval, as long as there was a reasonable likelihood of clinical benefit. But this, of course, is subject to agreement with regulators for each specific pathogen. And then if made available under accelerated approval, post-marketing studies would need to confirm benefit. And, you know, just to leave you with a final thought, I think influenza may be fairly well matched to the known strengths of mRNA vaccines. And if you think about what to do in an mRNA vaccine plan that is looking for things to do between making COVID vaccines, if one could make a flu vaccines there, for flu, the duration of efficacy, which I think is perhaps not yet quite well known for mRNA vaccines, is less critical. A real advantage of using mRNA vaccines for flu is that this might allow one to delay the strain selection until one had a better chance of matching the vaccine with the subsequently circulating strains. Right now, we're limited by the amount of time it takes to make in series 3 or 4 antigens of flu vaccines. And so we end up selecting our strains in February. But one could imagine flu vaccines, if they were made via this technology, being the strains being selected much closer to the actual beginning of the flu season. And so I had another one advantage, which I'm not thinking of now, but maybe will come out in the discussion. So in any event, this is how I've been looking at this. And so I would recommend that the South African plant at least start looking at flu. And for those of you who are interested in this, to see whether there are ways of adapting mRNA vaccines to influenza, which of course would also be very useful in helping us deal with another very important pathogen of pandemic potential. And I will stop there. Thank you very much.
And thank you very much, Phil. This was absolutely fantastic. So for me, what was amazing was that on both presentations, I really learned a lot. So I'm going to maybe, Phil, maybe just add one comment on flu. Maybe this was what you wanted to say, but this is what I've been thinking, which is an advantage of the mRNA is that we can produce also the nuclear protein, the neuraminidase, Because although we talk about H1N1 and H3N2, most of the vaccines that we give, we just look at the H. There's got to be a bit of N there, but we don't really quantify it. We don't really know what role it serves. And maybe now with RNA, we'd be able to get the N to actually have a real role to play. And we know that it does have some protective role. And we've been missing that with a lot of our vaccines. So I fully agree with you that the South African hub should, as soon as they've got COVID handled, they should start looking at influenza. So we're now going to go into the question session, and a lot of you have already been putting questions into the chat, and Barney has been very active already replying to some of these. But I'm going to begin already with the first one, which is Barney's replied in the chat, but Barney, I would like you to take this orally, if you can also. So this is from Andrew Lee. Andrew, can you put your camera on so that we can actually see what you look like? And the question is the type of inflammation and immune response induced by the mRNA vaccine, for example, interferon versus IL-6, is this important for vaccine immunogenicity? And how can this be influenced by the mRNA or antigen design? So Barney, of course, this question also comes from me, really. This is a question not only from Andrew Lee, but also from me. So Barney, can you put your camera on and can you take that question live?
Okay, I tried to answer this by typing, but there's two types of inflammation or immune induction I think you have to think about in terms of the cytokine milieu you're inducing. And they have an effect on reactogenicity and maybe also vaccine efficacy for different reasons. And so the first part is the innate inflammation that I was talking about that induces, that can just by the chemical nature of mRNA and the lipids, you can induce type 1 interferons like interferon alpha, beta, delta. And that also includes in IL-1 beta and IL-6 and things like that that cause inflammation. And sometimes those things are good. That's basically what we do with protein adjuvants to create inflammation to make a better immune response, adaptive response to the protein. But in the case of RNA, you want time to translate and make the protein. So you can reduce the amount of initial inflammation from the innate stimulus, you can express protein for a longer amount of time. And I think that has a better effect and better benefit than the innate stimulus that comes from that early chemical event. The other type of immune response that I think is important for RNA is that because you're making the protein in the cell, it has MHC class 1 processing that's going to induce CD8 T cells. And not only do you have the CD8 T cell, which is a cytolytic effector, but it makes an early gamma interferon response, and it basically forces CD4 T cell differentiation into type 1, TH1. And so that means interferon gamma and things like that, and TNF, which are more effector cytokines for clearance, and less IL-4 and 5 and 13 and 9, which make a lot of mucus and airway constriction and neutrophilic responses that are not that effective, and it inhibits cytolytic activity. The Th2 response has a lot of adverse effects, especially on a respiratory pathogen. And so I think you have to separate out the innate response and the adaptive response. They both have different ways of either affecting reactogenicity or efficacy or quality of the immune response.
Thanks, Barney. I'm going to just follow that up. So we know, for example, CureVac had an mRNA COVID vaccine, which really didn't give very good results. And we've also seen there was a self-replicating one from Robin Shattuck's group. Does part of what you've just spoken about, does this explain why some mRNA vaccines against the same antigen are providing protection and some of them are not?
Well, it's, I think so, but I don't know enough details to be very specific about those other products. I'm telling you what I think about the biology.
Okay.
And that is that in the manufacturing process, if you don't have ways of removing all the double-stranded RNA, you're going to get a lot of initial inflammation and reactogenicity. You might have dose-limiting toxicity that you just simply can't get over. And the adaptive response depends on how many cells get transfected. And so if you are transfecting a smaller number of cells because of, or they're being expressing protein for a shorter amount of time, you're not going to get a very strong adaptive response. And if you look at the overall efficacy of CureVac versus Pfizer, BioNTech versus Moderna, it really is a clear dose response curve. And CureVac only was able to give 12 micrograms. I don't know if that was a choice they made or if that was dose limiting toxicity, but the 12 micrograms falls right on the curve you would expect of about 40 to 45% efficacy. And then it goes up from there. If you can give a higher dose and if you can avoid dose limiting toxicity by either the manufacturing process or the lipid composition or whatever, and give a higher dose and have more cells transfected, you're going to get a better adaptive immune response that's going to be more effective. And so I think it's this balance that you have to find between reactogenicity and exposure to the protein in its right conformation for a long enough time to get over the activation threshold of the B cells. And so it's this balance between reactogenicity and the ability to express enough protein.
You know, can I just add to that, Barney? You may disagree with this because your knowledge of immunology is certainly superior to mine, but I think the fact that the CureVac didn't have the methylpseudouridine is a major factor in this as well, because if you just transfect RNA into cells, and as you say, you get double-stranded RNA, and you get RNA that then interacts with pattern recognition receptors in a way that the methylpseudouridine containing RNA doesn't, then what that does is it shuts down translation in the cell. And you can get as much RNA into the cell as you want, and you may just not get as much protein made. And so I think that it's probably multifactorial explaining what happened there. But I would place a bet on the methylpseudouridine modification being very important for translation of the exogenously administered mRNA.
I agree with that. I think it reduces inflammation, it improves translation, and but I don't think we've done a real strong head-to-head because there's also differences in the codon algorithms that they use.
For sure, yeah. So we don't know.
There's a lot that we don't know about secondary RNA structure that's not being accommodated in these vaccines, and I think having a lot more research on that and understanding how secondary structure is affected by codon choice and how it affects translation and efficiency, I just think there's a long way to go there.
So thanks for both of you answering that question. So I think what I'm certainly getting out of this is just because you've got access to mRNA technology, it doesn't mean you've got a vaccine.
Right.
You know, the methylpseudouridine, pseudouridine, the lipid, whatever, all of these can have an impact. And just because somebody else has been able to make a vaccine against that target and you've got mRNA, it doesn't mean that yours is going to work necessarily. So I think for everybody, bear that in mind, especially for those of you that are coming up with second generation products. So again, Barney, you answered this question, which is about mucosal delivery, which you answered very well, that we're trying to do this. There's a lot of research on there, but I'm going to actually throw this question over to Phil. Phil, the question about mucosal delivery of mRNA, how would you as a regulator see this?
You know, in general, regulators have been more relaxed about mucosal delivery of vaccines than parenteral delivery, just because it's less likely that the product is going to be distributed throughout the body. And so there are some, at least theoretical concerns that they don't have to worry about. Now, of course, intranasal vaccines are a special case of mucosal vaccines, where putting something up the nose is also putting it fairly close to the brain. And if there's a lot of inflammation associated with that, that could be a bad thing. And also there are a lot of nerves that pass by there. And so, so depending on the target, with the exception of intranasal vaccines, mucosal, for instance, vaccines against STDs might in fact have an easier time dealing with some of the issues that one would otherwise encounter. So I will, I'll stop there.
Okay, thanks. And Barney put a pretty good explanation a bit lower down in the chat for those who want to read it. So we've got a lot of questions coming in, so I'm going to move on. And there's one question here: how might regulators use RWE approaches to inform continued label expansion? Phil, Katie, over to you. And sorry, could Ferreira Katia put your camera on, please, so we can see what you look like? My apologies to Wellington for not having asked you to do so. Ferreira, if we can see you on the screen. So Phil, if you can answer the question.
I'm sorry, say the question again.
The question is, how might regulators use RWE approaches to inform continued label expansion?
So, of course, the difficulty is that you don't get RWE until a vaccine has been given in appreciable numbers to the people that you want the RWE on. And so it's a little bit of a chicken and egg question, is where you can't get the RWE if the label hasn't already been expanded, in which case the RWE can give you confidence that you did the right thing, but may not always give the exact answers you're looking for. But there are cases where I think there's a clear role. For instance, many people are giving vaccines to the immunocompromised, and we have sort of some idea that if you give vaccine to somebody with a moderate degree of immunocompromise, they might not respond to 2 doses. They probably will have a better response, for instance, if you give them 3, but there may still be a significant number that don't respond even to 3. And so as one can start gathering information about who's actually protected by a different number of doses and in a group like that, it seems to me like that's really very fair game for providing information on how the vaccines can be used. And so in general, for licensed products, the FDA has been reluctant to label RWE. I think under EUA, there probably would be less reluctance if the data were found to be compelling. And certainly, you know, we're using RWE to understand the efficacy of these vaccines in practice, potential waning of immunity, and so forth. And of course, a lot of the safety data on rare side effects is also all coming from RWE. So I think RWE is a big component of the decision-making process for a lot of things, even if one can't necessarily, for a number of these things, maybe one can't get the RWE one wants, and yet that doesn't mean it isn't playing an important role.
I'm getting a question, Phil. What is RWE?
I'm sorry, it's real world experience or real world evidence. So the idea there is that obviously data is collected post-marketing with the idea of trying to compare people who've been vaccinated with those who haven't, which is fraught with all kinds of potential problems and biases and confounding. And yet the people who do this are getting better and better, both at addressing the confounding and also noticing new sources of possible confounding. But nonetheless, it's in many cases, it's the best data that we have on important questions.
Okay, thanks, Phil. We're going to go again to a question that Barney's already put some answers in the chat, but Barney, I'd like you to go into this and also ask Phil to weigh in on this. So Michael Kawarik, if you can put your camera on. So the question here is speed. There's actually two questions. Speed to enter phase 1. What's the rationale that mRNA can go faster than other platforms? And isn't multivalency a limitation of mRNA due to the risk of adverse events if too much mRNA is used? So both Barney and Phil. Barney, over to you first.
Well, speed to me for RNA and to a lesser extent DNA is based on the fact that it can be chemical synthesis, and you don't have to establish the conditions for cell culture like you would for making a vector, or for making a protein, or for growing a live virus to either use or inactivate. So all those optimization of cell culture conditions takes time, and downstream processing takes time. And the vector-based approaches are probably second fastest to the nucleic acids, but making a protein in a cell line to make a protein can take months and months. You've seen this kind of play out with the coronavirus, that the mRNAs were fastest, the vectors were next fastest, the proteins are still trying to come along, and there was some rapid whole inactivated virus production, but to really optimize the cell culture conditions and grow a virus to a very high titer ordinarily takes a couple of years, and so this was done. It probably what was done can probably be optimized. So I think the main thing is the chemical synthesis and the fact that it's platform technology. The downstream processing is the same each time, and so I think that's the reason RNA can go faster.
That's a huge advantage.
You don't need a bioreactor. I was just going to say, you don't need a bioreactor. That's the reason it's a little faster than DNA.
Phil?
Although you do need DNA to start off with as a template for transcribing the RNA.
Yes, but you don't need as much as you would need. You don't need like 4 milligrams per dose for the DNA.
That is true. Yes.
Phil, are there any other speed ups other than sort of the absence of this need for the characterized cell line? Is there anything else that you can bypass? I imagine you still need to do your full your GLP tox study.
Well, no, Martin. At least for COVID, the mRNA vaccines were getting into humans before those studies were complete, based on an understanding of how other mRNA vaccines had done in tox studies. And so it doesn't mean that they aren't eventually completed, but in some cases where you can make a good case, and this is again something to discuss with each regulator in every individual case separately, but what one could make an argument for doing that in parallel, for instance, because one would have pretty strong evidence from previously done tox studies on something that is closely related, that they weren't needed. So, and that indeed was the case for the COVID mRNA vaccines.
But I assume that if we were not to go for a an mRNA flu vaccine, we would need to go through the traditional type of step-by-step approach.
I would not be so sure about that. Again, I'm a regulator only for a few more weeks, but nonetheless, the FDA, at least over a fairly long period of time, has really been thinking about identifying times when tox studies are not providing useful information, and very often does allow manufacturers to request waiver of tox studies because there's very strong evidence that we already know everything that those studies would have shown. It's up to the manufacturer, to the developer, to explain why it is that it's unnecessary, and to ask that. And yet, I think for an mRNA flu vaccine, given the huge number of humans who've received mRNA vaccines, the fact that these flu antigens are made in humans and are presented to humans and other vaccines, and people actually get influenza all the time, it's actually very, very hard to imagine that a GLP tox study of a flu vaccine would tell us anything that we don't already know. So I'm not promising that that's what a regulator will say, but I do think that that's an area where it's certainly not a certainty that such a study would be required.
Well, that's for many of us, I think that's going to be fantastic news if we can get our national regulators to follow that kind of thinking.
I mean, obviously, if one is looking to move quickly in a pandemic, if you want to do a tox study, that would slow things down quite a bit. So maybe one eventually does it, or one does it in parallel. But it seems like one could make the argument that one could start dosing people in phase 1 before one had that. But otherwise, would be a very limited amount of information.
Well, I think that could also reduce some of the cost hurdles that some developers face, because the GLP tox is quite often about the same order of magnitude as the phase 1 in terms of cost. So if you can actually delay that a little bit until you've got some indicative human immunogenicity, it might facilitate for many groups product development. There's a second part to this question about multivalency. Barney, is there a limit to multivalency?
Well, I've heard rumors that you can put as many as 20 genes in a formulated lipid nanoparticle and show that all of them are expressing. And so if the question is about multivalency and reactogenicity, I think there's a real question whether it's the dose of DNA or the RNA, which might be related to how much double-stranded RNA artifact there is in there or something like that, or lipid, because the lipid is proportional to the amount of RNA. But my sense is that some of the very consistent reactions that happen after the second dose of RNA that involve the local and systemic reactogenicity is often, it may be more related to the number of cells being transfected. Now, one of the reasons I think the vector-based approaches have more reactogenicity on the first dose and less on the second is that there just isn't as many cells transduced by the vectors because of anti-vector immunity or other factors. And so the number of cells transfected, if that's really the basis for that reactogenicity, and you're trying to reach dose-limiting toxicity, then having multiple genes in the same nanoparticle or multiple genes in the same cell won't really affect that, in my way of thinking. I think these are things that still have to be sorted out. But at the moment, I think there's evidence, at least in some of the early studies with things like CMV that have gone into phase 2 with Moderna, that you can express multiple proteins from the same lipid nanoparticle, and it doesn't really affect the reactogenicity. There are some problems of if you start putting multiple different class 1 fusion protein genes into the same lipid nanoparticle, and you want to make an HMPV and a RSV and a flu and everything at the same time. I don't think there's really any evidence of, or even 2 different coronaviruses. Do you get homotrimers or heterotrimers being formed, and how is all that working when the proteins can associate in different ways? So it doesn't seem to be an issue for the coronaviruses that were just tested in a mixed way, but that's one thing that might be of interest.
Okay, thanks, Barney. Phil, any comments to add to that?
Yeah, it's just obviously the more components you have, the more sure you would need to be that all of them are being expressed at the right levels. And so that may lead to some complexity. And if it were necessary to give much higher doses, then I think the jury is still out. We don't really know what the reactogenicity would be. Does that extra mRNA also then come with more lipid nanoparticle in order to envelop it all? And so I think this is very much a case-by-case thing. But I would say this idea of a multivalent vaccine is an added complication that requires people to pay attention to it.
Thanks. And I'd like to come to later to a question about biodistribution of these, but let's see if we've got time. So now there's a question to both of you from Shan Lu. Shan, can you put your camera on? So how much of the success of the COVID vaccines or the COVID mRNA vaccines is due to the fact that SARS-CoV-2 may be an easy target? And we all know that it's an easy target. Will mRNA vaccines, can mRNA vaccines induce similar levels of antibodies against other targets such as HIV? I think both of you kind of touched on this, and it's true that SARS-CoV-2 is easy, and both of you kind of said, well, it's not a panacea, but
well, but you know, I actually would take a little issue with that. If we didn't have the mRNA vaccines, and our best vaccines were of different modalities, at least the initial going made it look as though the AstraZeneca vaccine wasn't nearly as effective. It's unclear whether the Janssen is quite as effective as the mRNA vaccines are. Of course, the inactivated vaccines, the Chinese vaccines, the Sinovac, Sinopharm, are certainly not at the same level of effectiveness as the mRNA vaccines are. So I'm very, very impressed with the very high neutralizing titers that you get with mRNA vaccines. And I'm used to looking at vaccine immune responses and seeing those responses being generally lower than what you get with wild type viruses. And yet the mRNA vaccines are in fact inducing very strong humoral immune responses, which I think they are unusually high. And so I don't know that it's all just that this is easy, but we'll have to find out. But the critical thing, as Barney and I both said, especially for HIV, is I don't care what kind of vaccine you have, nobody knows what the antigen is supposed to be. And that's what's holding us back there. If we knew the right antigen for HIV, we would have an HIV vaccine by one or another modality, whether it's mRNA or not.
Barney, any comments?
Well, I do like mRNA for HIV because it can induce both antibody and a good CD8 T cell response. And so I think both of those things are going to be required to really have an effective vaccine eventually. But, you know, HIV antigen design has so many problems for antibody right now that I think we may have solved some of the conformational evasion problems, but that's only one aspect of all the problems that HIV has for evading broadly neutralizing antibody. So I just don't think like Phil said, we don't have the right antigen design yet or the right antigen composition. And you know, I think, you know, some of the things we're learning from influenza and some of the things we learned from coronavirus, from RSV, and I don't know, maybe we can go back around and get something and just have a heterotypic particle display or something that can somehow get some of these subdominant cross-reactive antibodies going. I hope so, but it's still a long way to go there, I think.
Thanks.
May I follow up just one briefly question?
Sure.
First, nice to see you, Graham and Barney and Phil. Good to see you guys. Thank you. Wonderful talk. My question actually was not phrased very well. I actually, was asking the relative immunogenicity rather than the pathogen complexity. I know HIV is a very different set of questions. I'm just asking the protection level, the threshold of protection. So look like this, SARS-2 virus, you have such a level, relatively low level neutralizing or binding, you can protect. The question is, we know the nucleic acid vaccine, the immunogenicity is relatively low. The question is whether this is good enough for some other viruses or bacteria. They need a higher titer. So Phil was right. Unfortunately, we did not have a recombinant protein vaccine as a control. Inactivated is a bad example. There are people, due to manufacturing issues, they did not make the SARS vaccine too here. But if those come out, maybe that's a good comparison.
That's true, but Miles Davenport has in fact put on his graph also at least Novavax vaccine where there may be some manufacturing issues. And yet the immune response certainly was no better than what we're seeing with the mRNA vaccines in terms of humoral immunity. And so I still would continue to say that we're doing pretty well with humoral immunity using mRNA vaccines as compared with other modalities. So I don't know that one is necessarily giving something up there.
Sure.
We've made CHO cell derived protein, and with some adjuvants, you do exceed what you can get with mRNA.
Yeah.
With the right protein and the right adjuvant, you can show in monkeys that you can get even higher. But the mRNA has really been remarkable for making antibody responses better than I would have expected based on all my experiences with DNA. And I think it's just because RNA works by getting into the cytoplasm and it doesn't have to go all the way in the nucleus. And so it just is more potent. More cells are transfected effectively with RNA than with DNA.
Okay.
Thank you.
Thanks. We will move on. I see there's lots of questions coming in, so I'm going to pick up the cadence a little bit now. So to Barney, so this is from Perraud Nicolas, if you can put your camera on. Barney, what about measles mRNA vaccine? Do you see a potential to have a better one than the current one, especially for early infancy protection?
Yeah, well, I think that's a great question. We've been working quite a bit on measles because the 40-year-old, 50-year-old manufacturing technology is, and a 40 or 50-year-old equipment is not easy to maintain. And so some of the manufacturers are struggling a bit to keep going with the current way of doing this. And so we may need a different type of measles vaccine. And the question is, does it need to be RNA or can it be a well-designed protein? Because at least for now, protein vaccines are a little more cost effective. And since we know what the antigen is, do we need an mRNA vaccine? We might need an mRNA vaccine for the reasons I mentioned about, worry about the right kind of T-cell immunity, and you may want a live virus-like priming or mRNA priming or vector-based priming instead of a protein priming. So the reason to do it for measles to me would be that you want a MHC class 1 antigen presentation priming event before you just start boosting up the antibody. And so for that reason, mRNA delivery of a good fusion protein or hemagglutinin protein would be, I think, a reasonable thing to do for measles. And then later you can boost it with the protein.
So obviously the downside there, the obstacles we overcome is we know that the duration of immunity with the existing vaccine is already very good. And the second dose is not a booster dose. It's really intended to catch the primary non-responders to measles. And so one might need to give kids 2 or 3 doses of an mRNA vaccine in order to duplicate what one gets with the live vaccine. And then you have the schedule getting more crowded. And then also, perhaps in this world where we're replacing lots of vaccines with mRNA, a lot more mRNA vaccines being given, and then perhaps questions then about the total dosing of mRNA. And of course, we don't really understand pediatric reactions to responses to mRNA vaccines yet anyway, especially if you get down to the age where measles is given. And so I think we have some questions to answer, which doesn't mean it's impossible. And I agree, it's an enticing target given given the immunology of this.
So thanks, both of you. And that actually leads directly, Phil, into a question addressed to you. And although you've got a few weeks left in the FDA, but so you might not want to answer this, but for you to decide how to do so. What is your opinion on why the Pfizer pediatric mRNA clinical trial was not adequately powered to detect myocarditis, at least at the most frequent level, for example, 1 in 6,000? I find it interesting the FDA asked Pfizer to increase the number of pediatric participants who vaccinated, but they did not increase to a level sufficient to address the safety signal. Now, you can't talk about your professional opinion here, but can you address this from your personal opinion?
Sure. I mean, obviously, when the FDA is looking at what to ask a manufacturer to do, they would like to get enough data to support a decision, and they would also like to get, but at the same time, they're mindful that they don't want to slow things down to the point where a vaccine that could have real benefit then is delayed from being made available to the people who would benefit from it. And so, you know, as the FDA was looking at the myocarditis signal, I think I'm imagining, from my personal perspective, I would say that, as you say, there were reports initially from Israel of myocarditis at around 1 in 5,000, and then other places reporting a little less frequently. But of course, this was age dependent, and what gave rise to perhaps more concern was what would be that it appeared that the myocarditis signal might have been higher in younger people, right, which would have been ages 16 and 17 versus older than that. And so the question then wasn't just, is the myocarditis signal the same in adolescents as it is in 16 and 17-year-olds? But of course, the question was also, can we get a sense of whether it's even worse, and what is one's threshold for even worse? And so there's never an exactly correct answer about what is the right number of subjects to have in a trial to address safety. And yet, I think there were a feeling was that a relatively small trial might not rule out concerning increases, whereas having a larger trial would have a better chance of catching it if there in fact were a higher risk in younger ages. And it seems like some of the real world evidence that is coming in now suggests that the risk actually isn't any higher, and might even be lower as you go down in age from that peak around ages 16 and 17. But just to give you a sense of how regulators might think about this, so it might not be, there may not have been a need to figure out exactly what the rate of myocarditis was in this age group, but there surely would have been some thinking as to at what point would the risk be so high that we would be really concerned, and we surely would want to have a chance of detecting that.
Okay, thanks, Phil, and my apologies to Julie for not asking you to put your camera on. We're going to go to the question again. This one's for Phil, regulatory from Wellington. Wellington, please put your camera on.
Hi, Wellington.
Yeah, you mentioned the importance of the lipid nanoparticle for mRNA vaccines. What is the current thinking from a regulatory standpoint of the lipid? Is this the active substance, the adjuvant?
So, you know, Wellington knows that FDA has wrestled with these kinds of questions for many years of how to describe adjuvants and so forth. It's clear that the lipid nanoparticle is playing some role, an important role in getting the mRNA into the cells. And so in that sense, it's playing that kind of a helper role. I would argue that to the extent that it's an adjuvant, it's also a helper role that would cause one to think of it as not the active ingredient of the vaccine, where the active ingredient, whereas the active ingredient clearly is still the mRNA, and you measure that response based on the immune response to the mRNA. But and I guess one way of looking at this is that one wouldn't market the lipid nanoparticle on its own as a delivery device which had some kind of a clinical outcome, but it really is only something that is meaningful when it's packaged together with the mRNA. So whether it's called an excipient or an inactive ingredient or whatever, some of this is designed to capture this idea that it's not active on its own, but its activity is only meaningful in the context of the full mRNA vaccine. So I don't know if that answers your question, Wellington, but that's how this is being looked at. Now, if over time it's realized that the lipid nanoparticle has clear adjuvant effects, and I stated my personal belief that it is causing some inflammation, which is increasing the immune response to the mRNA, then it may be that it would become classified as an adjuvant, but the FDA hasn't classified it as an adjuvant yet, to my knowledge.
Yeah, thanks, Phil. I mean, this is a tough issue, and I know that it may be evolving. So that's why I just wanted to get a most current update. And yes, thank you.
Wellington, thanks for the question. We've also been getting that question at WHO, and so I was very pleased to hear Phil's reply. The next question we have is from Sven Silverdahl. This one goes to Barney. Barney, about the dose schedule, I presume here we're talking about COVID. Why not have a dose 6 months after the first dose, either a second dose or as a third dose, because of the booster response? Isn't it true that it takes 4 to 6 months for a strong booster response? I'm not sure that I fully understand the question, but Barney, over to you. Otherwise, I can try and decipher it.
Well, I understand the question because I believe the same way that the 0-3 week or 0-4 week is really, in my opinion, a co-primary immunization. It's not really a full immunization schedule, and having a late boost 3 to 4 months later will really kind of set the tone for how all the B cell precursors are activated and made more durable. So I think really the speed or the rush to get something going, and knowing that you could at least get a decent primary immunization event with the early quick schedule, that's really why this happened. And Pfizer went even to 3 weeks with a 1 week endpoint after the second dose to save 2 weeks and catch up with Moderna. So I think a lot of this early dose scheduling regimens was really a matter of just rushing to get an answer. And I think you're exactly right that having a late boost is going to be part of this kind of vaccination in the future.
Okay.
So of course there is, I agree with Barney, is that there was a desire amid a country that really desperately wanted the vaccine to get the answer most quickly, and by giving the shots quicker and checking the immunogenicity results more quickly and so forth. This did speed up vaccine development. That was an important thing to do at that time. The other consideration, though, would be if one were to go to a 1-6 regimen, is if the single dose doesn't last that well through 6 months, you may actually be leaving people unprotected or less well protected in month 3 through 5. And so you wouldn't necessarily select the regimen which gives you the very best long-term response if you're trying to protect people in the middle of a pandemic either. And so when these products were authorized, of course, we still had rising cases, and we've had our wave since then as well. And so if there's a sense that giving the products closer together is better than giving it just, or giving the 2 doses closer together is better than giving a single dose, there still could be a reason to do that, even if in more casual times, one would prefer to separate them by more. And that then gets to this question of, well, maybe then to get the early responses, you do have to give the second dose at 1 month, but then that then pushes you towards thinking about the booster later on. Although thankfully, it turns out that those 2 doses were very, very impressively effective and still are, continue to save very, very many lives, even in people who've not been boosted.
Thanks to both. This question from Katia Ferreira. Katia, put your camera on, please. Not sure that I understand the question. Could mRNA solution for combination vaccines to reduce the need for multiple vaccines of a single target? I'm not sure, Katia, could you could you come on and tell us precisely what your question is? I don't understand that question. Okay, maybe while waiting, let's take the next question. Okay, this one is to Phil from Wellington again. Wellington, you're still on. We can still see you there. Will there have to be WHO and VRBPAC selection of seasonal flu strains first for the other platforms like LAIV and inactivated, and a later one for mRNA? Phil, over to you. I've got my opinion on this one.
So, I think the products or the public health mechanisms adapt to what is necessary in order to save the most lives. And so if it were clear that the mRNA vaccines could potentially be better matched by delaying the selection, I think there would be a very strong public health reason to do it. It would be very hard to argue against doing that. I believe that maybe not right away, but in a fairly short period of time, that process would adapt.
So let me weigh in there, Phil, thanks. I'm going to weigh in from my WHO perspective here, is that currently, for example, for the Northern Hemisphere, we make the decisions in pretty much February, March. Now, if we had to have another meeting in June to see how well this is progressing, well, that's just another meeting. But because we can get mRNA produced so much more quickly than the egg-based, I think there will come a point where we may have to be considering this, because this would also apply maybe to the recombinant proteins, the baculo-derived vaccines as well, which can also be produced quickly. So yes, I think one of the weaknesses of our flu program is the 8 months, 9 months sometimes, between when we make the decision on what the strains to produce are and those vaccines actually hitting the market. And we've got literally 1 year out of 2 or 1 out of 3 where we realize that things did not proceed as we thought they would, and we get the wrong prediction there.
Although we wouldn't always have known by June that that was the case, although we would have had a better chance.
Yeah, it's just a question of getting the odds on our side.
Yes.
But I think it's going to be very interesting to see how mRNA vaccines for flu, if we start using mRNA for seasonal flu, how we're going to be living with the mRNA vaccines and the classical vaccines on the market at the same time, and how these are going to pan out in terms of uptake. And I think
that finally reminded me of the additional point I wanted to make of why mRNA vaccines were something to consider for flu. And that is, unlike most of the other vaccines on my list, the seasonal flu vaccines aren't even that effective. And so there's a possibility that an mRNA vaccine, by being matched better, or perhaps by giving rise to stronger immune responses, could actually be substantially better. And so there should be a reason to want to explore that. Whereas if you're a developer and you're looking at a vaccine that is already 99% effective, and you're thinking, could we replace this with an mRNA vaccine? Well, you're trying to fight against an entrenched market that everybody's happy with and that everybody thinks works. In the flu arena, if you had an mRNA vaccine that were clearly more effective than the existing egg-produced vaccines on average, you would very, very quickly have an amazing market, which is another reason why I think that the flu is a good target for this.
But Barney, you've spent much of your life on flu. Any comments on mRNA for flu?
I think there's a great potential for flu, although, the hope, I think, for flu is that we can develop maybe a less expensive vaccine that would be more cross-reactive ahead of time. We wouldn't have to reformulate every year. But the place I see mRNA for influenza is, for instance, if you had the sequence of the new hemagglutinin, within 40 days or so, you could make, for instance, a ferritin nanoparticle with that exact hemagglutinin and deliver it by mRNA and have a a specific, vaccine for that strain of flu that should be extremely effective. And so it would be a much faster, you could probably beat egg-grown influenza vaccines by several months, which could make a big impact on the overall pandemic. And so to me, the value of mRNA for influenza might be in seasonal flu, but I think it'd be more likely to be helpful for a response to a new pandemic strain where you could make a specific product quickly.
Okay.
So let me just add to that. I mean, and this is, of course, maybe too futuristic for us to contemplate right now because it would require substantial adaptations by regulators as well. But one could imagine if one had a 40-day lead time to manufacture vaccine or whatever was 60 days, is continuously updating the strain that was in a vaccine to best match whatever is circulating. And so it may not be just a question of having a meeting in June to decide what is the mRNA vaccine for the following season. But if one knew in November that one had missed, by January, one could actually be giving a vaccine that isn't a miss. And so one could imagine doing real-time updates to the vaccine to keep up with the evolution of the virus. So again, that's not how this is going to start, but that's another possibility that mRNA gives you in seasonal flu that obviously the standard methods don't.
I think a lot to learn about flu because of the pre-existing immunity and whether you can do this as a membrane anchored, individual trimer or a secreted protein or a secreted particle, or I just think there's a lot to figure out there to how to best use RNA for flu.
So I would like, we could have a seminar just on mRNA and flu. My personal view is really lovely to go down to very young children, to infants, and give them essentially their first exposure to the antigens. So giving them the clade 1 and the clade 2 and the B early in life, and see whether that's providing long-term protection against severe disease, which is one of the hypotheses suggesting that it's your first exposure that predicts how you're going to respond to flu later on in life. But let's move on. There are many questions. There's more and more coming in. So this question now from Elizabeth Menkem. Can you put your camera on? Each individual sometimes have more than one vaccines. Are there interactions among the different vaccines? Is it positive or negative to the individual, especially children? Phil, over to you.
Well, this is studied very intensely when vaccines are given at the same time in the same visit. Very often there are concomitant administration studies to determine whether there are increases in adverse events, which is usually not the case, or decreases in the immune response to one vaccine or another. And a classic example of this is the MMRV vaccine, where it turns out that if you give measles and varicella together, the measles probably induces enough of an interferon response that the varicella virus and the varicella component of the vaccine doesn't replicate as well. And so in order to give the MMRV as a single vaccine together, you actually have to give more varicella in that combined vaccine than you would if you were giving this as separate doses. But many vaccines are almost all vaccines in pediatrics, they essentially all are, where two different vaccines might be given on the same day. There are studies looking to make sure that there isn't interference between the immune responses of one vaccine on the other. And so that is a serious concern, which is addressed in vaccine development.
Thanks, Phil. I'll just add to that currently there's a lot of studies on co-administration of influenza with COVID because the population, the prime population targets are the same. And if there's any seasonality, it's likely to be the same. And the data for the moment suggests that there is no negative interaction. A question to Barney. So Idoko Buki this is from Henry Dex. can you put your camera on? So the question to Barney, can you quantify or explain the expected mRNA vaccine stability progress, thinking of the cold chain in the low and middle income countries? What's happening in terms of stability?
Yes, well, Phil could probably say this better than me, but you can only use your vaccine as it's been tested and proven to work. And so, at the beginning, Moderna had taken this a little further than Pfizer BioNTech, and so they could have a minus 30-degree storage. And Pfizer, that had the same vaccine, had to use minus 80. But eventually, this is going to be lyophilized in room temperature storage. So there's a lot of progress going on to get the mRNA lipid nanoparticle into a lyophilized state where it could either be reconstituted and injected, or maybe applied to a microneedle, or all sorts of things. So I think eventually the stability issues will be solved. They just haven't been proven yet. So maybe Phil has more to say about that.
Well, vaccine manufacturers put a lot of effort into formulation and figuring out what buffers you need in order to keep a vaccine stable for as long as possible. And there are examples in the vaccine world of manufacturers that didn't do that. It ended up getting extremely short dating periods because their vaccines aggregated in the buffers they were using. I'm not naming any names. But then there are other manufacturers that are very cautious about this and test many different buffers to figure out what needs to be used in order to maintain stability. Because it's not just will the mRNA degrade. In fact, I think methylpseudouridine containing mRNA is certainly much more stable than native mRNA. But it's also how will this, stay appropriately encapsulated in the lipid nanoparticle? What possibilities are there for aggregation or perhaps even fusion of lipid nanoparticles to make larger particles over time? All kinds of things, especially as temperature goes up also. And so these are all studies that need to be done. And as Barney said, in the early pandemic, nobody had the time to do studies to figure out what is the best formulation, or indeed even what higher temperatures could we get away with storing these vaccines at. But this is going to be a topic of intense interest. And I have no doubt that we'll have mRNA vaccines eventually that have much more favorable storage conditions than what we have right now.
Thanks, Phil. So next question is from Idoko Buki. Can you put your camera on? And I think we might already have addressed this. Is it possible that the failure of the CureVac candidate may also be linked to the lipid nanoparticle? So we've spoken about the absence that they were using a non-modified nucleotide. I don't know what lipid they were using. Barney or Phil?
I think that that's the reason it's so hard to compare the CureVac result to the Pfizer result, because it could be the codon modification algorithm, it could be the lipid nanoparticle, it could be the contaminants of double-stranded RNA or whatever else is in the formulation, or it could be the 1-methyl-uridine or pseudouridine, or the dose. It could be all those different things or a combination of those things that made it work less well. But based on what I see from their data, the level of immunogenicity they had is very similar to the dose level of immunogenicity if you'd only use 12 micrograms of one of the other RNAs. And you could almost draw on Miles Davenport's curve, the level of efficacy they achieved based on their immunogenicity. And so I think it's largely dose, but it could be a lot of all those other things.
Okay, thanks, Barney. So we're getting close to the end, and so I'm going to take one more question, but this one I'm going to take myself. from WHO, and this one is from Nadja Kurioboulos. You can put your camera on. The question is, how can regions that have not yet started making vaccines for their population benefit from these great advances and make vaccines for their need? One such region is the Arab countries, making up more than 450 million. So the answer here is that WHO, with its partners, Medicines Patent Pool, has established a technology transfer hub specifically to enable each region to have the know-how to make mRNA vaccines, and then soon there's going to be we'll be setting up one for other types of vaccines. And this weekend, WHO will be launching a call from four countries that are interested in acquiring the know-how to respond to that call. So the technology is being set up. This is going to then be able to transfer the technology to regions such as for us, the Eastern Mediterranean region, which is where most of the Arab countries are. And so that each region the objective is that each region will be able to respond to an outbreak autonomously. But so I'm going to stop there. Phil and Barney, thank you both very much, but I'm going to ask each of you, would you like to make a final statement, a final comment about where we are going with vaccines? So, philosophically, Barney, you've got many years behind you and hopefully many years still ahead of you. Where...
You know, what I can say is this is a very exciting time to be involved with vaccines. The technology is improving. People are understanding the importance of vaccines. The focus on vaccines that we've seen now over the last 2 years has been unlike anything I've ever seen. And it's clear that vaccines are life-saving products that make a huge difference. And so for the young people in the audience, I applaud their career choice and their interest in this. This is an amazing time with a lot being learned, a lot more to be learned, and we need smart people who are going to figure all this stuff out.
Thanks, Phil. Barney, final comment?
Yes, well, like Phil said, I mean, the number of efficacy trials completed with different modalities creates an unprecedented database that's going to be looked at for years to come, and we still don't really know. We're going to learn all the different properties of these different vaccine modalities because of these multiple, somewhat harmonized with endpoints and assays, all these efficacy trials. But what I want to say is that the reason I retired from the federal government in large part was because I want to spend whatever time I have left on moving this kind of technology into low and middle income countries and then into regional, so it can be applied to regional problems. And so the question about the Middle East, you know, my question right now is why haven't the healthcare providers at all the hospitals in Saudi Arabia who are still at risk of MERS coronavirus, why hasn't there been an mRNA vaccine expressing the spike of the MERS coronavirus made available to them? These kind of regional problems, I think, are susceptible to this small batch, small footprint type of manufacturing that mRNA offers. And I would love to see the healthcare providers in Saudi Arabia have access to a MERS vaccine. These kind of things, I think, can only be solved regionally, and it's up to the global resources to try to help make that happen.
Can I say anything?
We've got 0 minutes left, so I'm sorry, I'm going to cut you off there. I'm going to thank Phil and Barney, but I'm going to pass the back to Philippe Duclos, who leads ADVAC.
Okay, well, thank you very much. Thank you on behalf of ADVAC. I mean, we had a terrific session. Thanks to you, Martin, for this great sharing and our outstanding speakers. I mean, clearly you are passionate, and what you were saying, Barney, at the end about investing in the further development and translation of the technology to LMICs is definitely great to hear. So thanks for your energy. thanks to all the participants, the many questions and the comments that were made. Sorry, we don't have more time. Actually, clearly, this could generate, as Martin said, many other webinars. Just to tell all of you that our next webinar will be on December the 8th. And will be on the safety of COVID vaccines. And I hope also we'll get a great panel of speakers. And I want to just finish with thanking Carol Berriesville, who is helping organize these webinars, and our technical support company, Artcast, for their great work also with the organization. So thank you, everybody, and have a good morning or good evening wherever you are in the world. So thank you. Bye-bye.
Thanks
Thank
very
you.
much, Barney.
Great to see
Thanks,
you,
Phil.
Barney.
Thanks, Philippe.
And hopefully, again, as I said, everything will be posted as of tomorrow, very soon on the ADVAC website. So thank you. Bye-bye.
Thank you.
Thank you.